What if the key to treating the deadliest brain cancer has been hiding in plain sight all along? If you believe the news, scientists have just found a breakthrough, but such clickbait has been around for many years. Let's explore what's really behind the concept of drug repurposing and glioblastoma, probably one of the worst diseases you can imagine. As always, we will cover nice insights across chemistry, biology, and medicine. Check this out. The same drug that kills cancer cells can be used in the chemistry lab to decorate molecules with cute little triangles. But beware, 50 years after its discovery, we've only recently realized that it's actually a class 1 explosive. So, discovering a new drug often takes more than a decade and billions of dollars in cost. Is there a simpler way? Drug repurposing uses a drug that's already been approved for one condition and tests whether it can be used to treat another. There are obviously limitations to how many indications a given drug may treat, but the nice thing is that we already know that the drug is relatively safe and we can save a lot of time and money by running a shorter research and development program. A long list of drugs have been repurposed, particularly against cancer. For example, loyal viewers might remember the crazy story of thalidomide. Originally used to combat morning sickness, it was pulled off the market due to causing severe birth defects. 3 decades later, it was found to be very effective in unresponsive multiple myeloma, a type of blood cancer. Probably the most common example for this is Viagra, originally studied in cardiovascular diseases due to its blood pressure lowering capabilities. The discovery of an unexpected side effect triggered Pfizer to instead develop and commercialize it in ED. They even repurposed it, as low-dose Viagra was able to help treat pulmonary arterial hypertension, a very serious disease with few treatment options at the time. The opposite happened for dimethyl fumarate. Here, the drug previously used for treatment of psoriasis, a skin disorder, was ultimately developed in a higher dose for the treatment of multiple sclerosis. The drug sold for a total of $3 billion in sales per year at its peak. So you might argue that companies are only happy to repurpose drugs if they can also make a buck out of it. But there's a limit on what types of drugs we can repurpose, particularly as we look into brain cancer. You see, the brain is wrapped in protective layers of cells. There are many, but the most critical one is the one separating the brain's bloodstream from the rest of the body. Without it, just one example, you would have permanent infiltration of inflammatory substances which would wreck the fine-tuned microenvironment of neurons. Disruption of the blood-brain barrier is related to neurodegeneration and dementia, so obviously we need it. The downside is that it restricts the delivery of larger molecules and hydrophilic drugs, so many agents that might be effective against brain cancer would not be able to go where they're needed. The conclusion is that we should try to repurpose neuroactive drugs which are already known to be brain penetrant, like antipsychotics or antidepressants. But why should we look for new drugs for glioblastoma? Well, this aggressive brain cancer has extremely low survival rates, can't be prevented, and it's so invasive that it rapidly spreads across the brain. Research shows that unless at least 98% of the tumor is removed, surgery is rather ineffective. I can't put it differently, this disease is a real piece of crap. It originates from the glial supportive tissue in the brain's white matter, but we don't know much about its cellular origin or risk factors. One key issue is that it also manifests in different forms and the tumor environment itself is also highly heterogeneous, hence also the name glioblastoma multiforme or GBM. Symptoms are bad, including seizures or memory loss, but they are non-specific and in many cases it's only diagnosed after the cancer has grown significantly. Compared to other solid tumors like breast cancer, we do not have an armamentarium of drugs. Once first-line chemotherapy treatment in combination with surgery and radiotherapy fails, we're just grasping for straws. It should be obvious to you why we need more medicines here, but let's learn about the main chemotherapy, temozolomide, to understand how dire the situation really is. The drug development effort for GBM started in the late 20th century and amongst other focus areas, looked into nitrogen-rich compounds like this one. These readily cross the blood-brain barrier, but the first-generation drug triggered severe bone marrow toxicity in patients, so it did more harm than good. As it turns out, the chlorophyll group is the culprit. By swapping it out for the methyl group, a relatively non-toxic and efficacious drug was born. By the way, if you're a chemist, you might assume that the nitrogen-nitrogen bonds in the ring might want to go kaboom. You're not wrong, but this one is really shocking. Previous literature has claimed stability at over 200 degrees, but only in 2021 did chemists discover it can violently decompose at much lower temperatures already. This is not an issue to patients taking formulated pills at room temperature, but large-scale manufacturers of TMZ maybe should know that it's a class 1 explosive. The paper is open source, so I invite you to take a read. It's pretty hilarious to see how you would go about classifying an explosive. One way is to drop a weight on the substance from varying heights. For TMZ, already low impact energies led to a decomposition and smoke, which is not great. But TMZ didn't flunk every test. You can't really tell from this picture, but burning 100 grams did not lead to an explosion, so that's a pass. There's probably a funny rabbit hole of experimental publications on explosives to be explored. However, the next figure is not funny at all. TMZ on top of radiotherapy does provide a significant overall survival benefit to patients with glioblastoma, but look how steep the curves are. TMZ extends median survival by only 2.5 months. The stats are honestly depressing, but there's more than meets the eye. Check out these two graphs, differentiating outcomes for patients based on so-called MGMT methylation. Learning more about this will teach us a few things about chemistry and biology. Before we get into the nitty-gritty, I wanted to express my gratitude to all my channel members and patrons. Some of these nerds have supported me for over a year, truly insane. But also, thank you for watching and being interested in these videos. So to understand TMZ's mechanism of action, it will help to know how TMZ is synthesized in the first place. If you have a chemistry background, you might want to pause the video and ponder for a moment. The key synthetic task is the linkage of the 3 successive nitrogens. The first step converts the amine to the diazonium ion. You commonly see the N2+ function as a leaving group in aromatic substitution reactions, but here it reacts with methyl isocyanate in a dipolar addition, creating the crucial NN bond in a spirocyclic intermediate. Finally, a 1,5 sigmatropic shift rearranges to the aromatic system and gives temozolomide. Its mechanism is really cool and similar to other cytotoxic agents that modify DNA bases. Do you have any ideas? Basically, we are reversing the 2 synthetic steps, but with a twist. Already at normal pH, water molecules in the body can open up the ring system and trigger a decarboxylation. This is close to a reversal of TMZ's second synthetic step, the dipolar addition with isocyanate. It's usually said that TMZ is a prodrug of this active form of the drug. In humans, the half-life of this decomposition is roughly 2 hours. But it's not over. The nitrogen-nitrogen bond can be broken, which reverses our first synthetic step, the diazotization. As a result, a methyl diazonium ion is cleaved off. So in some sense, TMZ is actually the prodrug of this elusive species. This is CH3 bound to nitrogen, an excellent leaving group, so it's happy to react with nucleophiles. Because DNA is full of base pairs with nucleophilic sites, it is commonly the victim of such electrophilic alkylating reagents. The reaction with guanine proceeds very fast at the nitrogen here, but we will see that this pathway is not as dangerous to the cell. Instead, the transfer of the methyl group to the oxygen is more problematic. This is because the O6-methylguanine product does not behave like a normal guanine should. Most will know that DNA connects with other base pairs through hydrogen bonds. Guanine likes to connect with cytosine. O6-methylguanine does not have the same contacts. It lacks the NH donor, and the methylated oxygen is a much weaker acceptor compared to the normal carbonyl. Because the match with cytosine is not great anymore, another pair can be formed instead, this time with thymine. This restores one of the 2 missing hydrogen bonds, so the second order impact of guanine methylation is that the DNA starts incorporating different base pairs. As already mentioned, the different DNA base pairs have many electrophilic sites and can generate up to 13 different alkylated unnatural products. The O6-methyl product accounts for only 8% of the total alkylation, but it has the greatest potential to kill the cell. This is because the unnatural pair with thymine represents an unsolvable problem to the cell's repair machinery. For reasons we will not explain, the mismatched repair mechanism triggers continuous, futile cycles of attempted repair, eventually leading to DNA strand breakage and cell death. The cell has one trick up its sleeve, a methyltransferase enzyme called MGMT. To remove the unwanted methyl group from the oxygen, we need a very potent and soft nucleophile. Such cases call for the thiol group of the amino acid cysteine, which can regenerate guanine through a SN2 substitution reaction. The twist is that while every normal cell expresses or produces MGMT, 60% of glioblastoma cases do not. In these cancers, the MGMT gene providing the blueprint for the enzyme is modified. Don't get confused because unfortunately it's again a methylation of the gene that silences MGMT. Now we understand the two different survival curves. The lucky share of patients with a methylated gene have tumors that lack the enzyme, explaining the high susceptibility to temozolomide. Unfortunately, patients with tumors that express MGMT as normal see no real benefit from the chemotherapy. We really need options, particularly for these MGMT negative patients. Let's wrap up this chapter with 3 final insights. The first is very logical. If one MGMT enzyme can only demethylate one O6-methylguanine, can we dose temozolomide so high that it even harms the non-susceptible tumors after fully depleting MGMT? Although this hypothesis seemed promising from animal studies, clinical trials in humans looking at dose-dense regimens have not shown superiority. In fact, on this chart you can see that a much longer 21-day regimen was even worse than a 5-day regimen. More is not always better. Our last 2 learnings are more chemistry related. The first one is the exploration of TMZ analogs that might be efficacious despite the presence of the MGMT transferase, the glioblastoma savior. Here you can see that this slightly modified version dubbed CPZ leads to more cell death compared to TMZ. But what's more is that even in MGMT negative cells, the potency remains high, while for TMZ it drops off significantly. The difference is that CPZ releases a propargylic diazonium ion, which can act as an electrophile not just once, but twice. The authors postulate the mechanism 1 is most likely, consisting of a SN2 prime substitution to give an alanine DNA intermediate. This species is still electrophilic, so other nucleophilic bases can add to it, creating crosslinks. The cell dies if it has more than roughly 20 crosslinks, and this mechanism can't be reversed by the MGMT transferase. This research is very early, but it's pretty cool to see how creating a different diazonium ion favors a different mechanism of action. Speaking of the diazonium ion, because it is released by temozolomide, organic chemists have recently figured why not use the pharmaceutical as a diazomethane reagent for synthesis. From a handling and curiosity perspective, it seems nicer than some of the other diazo reagents we have available. As we've seen earlier, the claim that it's non-explosive did not age well. But in my book, blowing up is not such a big deal if you can do some innovative chemistry in your last moments on Earth. Beyond simple esterification reactions, they found TMZ also cyclopropanated aromatic alkenes under iron catalysis. The yields and scope are not great, but quite cool that we can use the same species that kills tumor cells to make cyclopropanes. After having learned more about TMZ than you ever wanted to, you are wondering what this new research is about. In the spirit of drug repurposing, which we've covered at the start, the researchers try to find known neuroactive drugs that might be able to target weak spots in the cancer's biology. To identify such hits, they used a screening platform that leveraged imaging techniques as well as computational methods. Despite the fancy appearance, this is the real deal. They processed fresh tumor tissues obtained from glioblastoma patients undergoing surgery and screened them on the platform. Simply put, they isolated tumor cells from patients, incubated them with a given drug, and stained the cells. Based on how many stains stick to the sample, they calculated a specific score to assess the relative amount of cancer cells. The higher this score, the more effective the drug at specifically killing tumor cells. Here's an example that will explain the logic using TMZ, the drug we know well. The dots represent the PCY scores after incubating a patient's sample with TMZ. As we've learned, some patients are positive for MGMT methylation while others are not. This means that cancer cells in the green group on the right have the privilege of being able to reverse TMZ's alkylation mechanism. This group has an average PCY score that is significantly lower than the one on the left, and a lower score means more tumor cells and less benefit from the drug. The imaging platform produces beautiful matrices like this one. From the left to right on the x-axis we have different known drugs. Their effect in terms of PCY score is mapped across all patient samples on the y-axis from top to bottom. You see that drugs in the middle do not elicit massive PCY scores, but on the left we can see that some drugs have consistently dark blue boxes across most patient samples, indicating a high PCY score and thus strong anti-cancer activity. Aggregating across all patients reveals top and flop drugs. The top 4 drugs in blue are not surprising. They are anti-cancer drugs that act by stopping cell division, for example. However, the next best hit was the neuroactive drug Vortioxetine, showing significant efficacy in 18 out of 27 patient samples. On the other extreme, we have paliperidone with the most negative PCY score. This drug treats schizophrenia and it seems that it has higher toxicity to non-tumor cells in this assay. Vortioxetine is an antidepressant that can cross the blood-brain barrier and inhibit the serotonin transporter, but it does so in a weaker manner than the well-known SSRI drugs. On the flip side, it has a unique profile due to its strong affinities to 5 other serotonin receptors. The researchers ran other experiments to elucidate Vortioxetine's mechanism, but we will skip these and instead wrap up with how this hit translated from the test tube into a living organism. For this, they implanted human glioblastoma cells into mice and compared how well Vortioxetine and other neuroactive drugs performed versus Temozolomide, the green line. TMZ prolonged survival the longest, but among the neuroactive repurposed drugs, Vortioxetine in purple was most effective. You can also see that paliperidone, our worst PCY hit, did not perform better than negative control without any drug. The real promise comes from this chart. By combining Vortioxetine with temozolomide or another alkylating drug, the median survival increased by 20 to 30% compared to the single agents. You can also see the negative control had no survivors after roughly 45 days, while the combination arms showed long-term survival of a few mice. The normal caveats apply. This is a small sample size of mice. But the complementary mechanism is very promising, so the researchers are preparing a clinical trial to investigate it as an add-on therapy in humans. All in all, there's hope that this safe and cheap known drug will extend survival by a few months, but obviously it will never be a cure. A last promising signal comes from past research analyzing real-world electronic medical records. They looked at patients who were prescribed fluoxetine or Prozac, a different antidepressant, while also getting treated for glioblastoma. In these 10 patients, survival was longer, not a controlled trial but still interesting. If we take a last look at the drug ranking, we do see that fluoxetine has a positive PCY score. Because Vortioxetine is even more potent, you could hypothesize that it might work even better. However, this does not call for self-medication with Vortioxetine or other antidepressants. Some evidence also shows that antidepressants are associated with worse survival in glioblastoma patients. The reasons are unclear. It might be that the depression itself reduces survival, or it might be that biochemical mechanisms of some antidepressants are worse than what we've seen for Vortioxetine. All in all, science is definitely complex. Let's also give an honorary shout out to Mibefradil. This drug was initially developed for high blood pressure as well, but there were hopes to repurpose it for glioblastoma. Early studies showed that its unique mechanism of arresting the cancer cell cycle enhanced the effects of TMZ and radiotherapy. Unfortunately, I think it didn't progress through the clinic due to safety. One of the many approaches that might be more transformational is CAR T. Here, scientists engineer a patient's T cells to recognize specific receptors on cancer cells, transforming them into targeted killers. This technology has been successfully used in different types of blood cancer, but now there's also early research on bivalent CAR T in glioblastoma. Monovalent CAR T, so ones just recognizing the EGFR receptor, for example, have shown only limited efficacy in humans. The idea is that by binding to two targets, the killer cells have a higher likelihood of knocking out the cancer cell for good. One recent phase 1 study of such a bivalent construct reported significant reductions in tumor size, giving some early validation to the concept. CAR T cells can trigger major immune response and neurotoxicity, so evaluating the safety of these therapies is key, particularly when you inject them directly into the brain. So far it's looking fine, and because glioblastoma is such a horrible disease, the threshold of acceptable adverse events is relatively high. Thanks so much for your interest in this topic. If you liked it, make sure you watch my other videos as well. Let me know if you have any questions or suggestions for future videos, and I will catch you in the next one.